Students, in this class we are going to learn some of the important diseases of citrus crop. The most important disease that affect the citrus crop that includes gummosis or leaf fall or fruit rot disease caused by Phytophthora citrophthora as well as Phytophthora palmivora. So this affected the disease affected plants in the disease affected plants, a sap that oozing out from the small cracks in the infected bark, which is giving the tree a bleeding like appearance. The profuse accumulation of this gum from the bark of the trunk in the affected trees. The bark stays firm but dries and eventually cracks and sloughs off the appearance, giving sloughs off appearance, that is removal of cut off like appearance. The lesions which spread around the circumference of trunk and slowly causes girdling of tree. The affected tree may decline rapidly within a year after infection. This is the typical bleeding symptom that appears at the base of affected crop, and this is the foot rot symptom established by Phytophthora citrophthora or Phytophthora palmivora, and this is the typical gummosis exudation of oozes from the cracks. And this leaf fall and fruit rot symptoms are severe in mandarin oranges during heavy rainfall especially in South India. Development of water-soaked lesion at the basal portion of the leaf and extend to the whole part of leaf. Finally, the affected leaves may drop off. Similar water-soaked patches on the fruit rind also. Dropping off of the fruits these fallen roots they completely rot and they are with the profuse white mycelial or cottony growth on the fallen affected fruits which emit a foul odor or smell. So as I told you, this disease is very severe in mandarin orange, especially in southern part of India. This picture shows the severe leaf fall symptom, and this one shows the initiation of fruit rot symptom. These fruits may fallen on the ground, and upon the fallen fallen and rotten fruits, you could see the white mycelial growth. The lesions on the leaf bases especially ultimately leads to blighting and dropping of leaves. Upon the fruit, water-soaked lesions formed on the rind and responsible for complete rotting of the point of attachment resulting in dropping of fruits. The fruits lying on the ground with the mycelial growth. The causal organism, that is Phytophthora citrophthora as well as Phytophthora palmivora, which is known to produce sporangia during asexual reproduction, and the internal content of sporangia are differentiated into zoospores, and the sexual spore here is the oospore, and it also produces chlamydospores. It also produces chlamydospores. In the pathogen that grows on the fallen fruits, in the infected twigs as well as on the leaves and upon the cracks developed on the tree. The irrigation water, rain splash as well as the wind as well as the insects which are responsible for spreading the disease. Sweet oranges and acid lime and lemon are highly susceptible to this disease, and heavy soil that is what I can say like black cotton soil which is having poor drainage capability, and high water table, high soil moisture, pH ranged from 5.4 to 7.5, and the temperature of 25 to 28 degree are responsible for the development or helping or favoring the development as well as spread of the disease. This is the typical pear-shaped sporangia produced by the Phytophthora, and upon maturation of this sporangia, internal contents are cleaved into zoospores, and these zoospores are released through after opening of the sporangium, and this is the initiation of sexual spore formation. And the sexual spore whose spores are dark brown in color, double walled, thick walled, and which may act as a resting spore. In order to manage this disease, we have to use some of the resistant rootstock like sour orange, Cleopatra mandarin, etc. And then those soil which are having poor drainage system should be avoided, and avoid excess irrigation water also, and avoid continuous contact of the stem with the irrigation water. That is, excessive irrigation should be avoided. Right, and then scrape out the affected point and disinfect the area with the mercury chloride or potassium permanganate using a cotton swab. And then this planting pits should be dusted with the zinc sulfate and copper sulfate as well as lime in the ratio of 5:1:4, and painting of Bordeaux mixture or Bordeaux paste on the tree trunk up to 5 to 75 centimeter above the ground level once in a year, which also helpful in development of this disease. Soil drenching of Bordeaux mixture 1%, or difoliton 0.3%, Dridomil 0.2%, or Alliate 0.2% are also recommended to manage the disease at field level. And the second thing is the Diplodia gummosis, which is the dry gummosis. The wood and the trunk of large branches are highly affected, and this pathogen is also affects very seriously the sweet orange as well as the mandarin oranges. Due to the infection, the gummy oozes are exuded from one or two spots near the forking region, that is the branching region, or the bigger limbs. And then the infection starts at the crack on the branches and spreads to the above ground level. The infection which cause growth cracks or ridges on the stem of affected trees, and the bark is also got killed due to gummy exudation and dries up, and finally, black exudation may come out from severely affected or severely infected trees cracks, and this is the gummy exudation due to the dry root rot, that is Diplodia natalensis. And this Diplodia natalensis is known to produce pycnidia, which is a flask-shaped structure, a rampant and papillate structure, and which in turn contains conidiophore and conidia. Here the conidia are initially single-celled hyaline, and at the time of maturation become bicelled, elliptical, brown in color with a lot of striations on the outer surface. Wind as well as the rain splash helpful in spreading the conidia. Reduced tree vigor as well as the damages caused by the insects, poor nutrition, and age of the crop are also responsible for development and spread of this disease. The wounds caused on the bark, especially on the limb as well as on the forking region, should be scraped out and protected with the Bordeaux paste. The broken limbs should be should be removed, and the affected portion must be cleaned, and the cut end should be protected with Bordeaux paste or Bordeaux mixture, etc. And then next important disease is powdery mildew, which is caused by Oidium tingitaninum. Earlier it was known as Acrimonium tingitaninum, and this is Oidium type, so it produces white powdery patches on the upper surface of the leaf. And as we learnt in the mango crop, here also you could see the powdery appearance or powdery patches on the young leaves, and they are young flushes including the inflorescence, and those are covered with white powdery patch and later turn into dusty gray. And the ends of powdery mildew affected leaves are twist curled upwards and malformed. Severe infection ultimately leads to drying and defoliation. Upon the fallen leaves, it produces the sexual ascocarp, that is Chasmothecium, which in turn contains ascus and ascospores. So ascospores, which serves as primary source of inoculum, and then the airborne conidia are responsible for the secondary spread. And this is the typical white powdery patches, especially on the young flushes, young fruits and inflorescence. Severely affected plants show severe defoliation. And this is the type of conidia produced by the Oidium tingitanium, that is, it produces short stout conidiophore, and which bears chain of barrel-shaped conidia arranged in a basipetal succession. And as we discussed, the sexual ascocarp, that is Chasmothecium. This is the typical Chasmothecium with myceloid appendages. So to manage this disease, apply sulfur dust very early in the morning or spray 0.3% of wettable sulfur at fortnightly interval. That is highly advisable to reduce the powdery mildew incidence. And the next important disease is scab or varicosis. Scab or varicosis, which is caused by Elsino fawcettii. It starts as a small semi-translucent spots which finally become sharply defined pustular elevations, especially on the undersurface of the leaf. The affected leaf tissues become distorted, wrinkled, just opposite surface or corresponding to the warty growth that shows circular or pinkish discoloration with the red center. Similar type of spots may also be produced on the twig. The crust of these growth become covered with scabby, corky tissues. Upon the fruits, it produces lesions that consisting of corky projections, which often break into scabs. These fruits become rough and become completely distorted, and affected fruits may fall off. And this is the scabby growth on the rough lemon and tangelo, etc. And Elsino fawcettii or Sphaeciloma fawcettii is known to produce more acerulae, and the acerulae in turn contains conidia, conidiophore, and setae. These conidia, which are hyaline, oblong or elliptical with two minute oil globules at the ends, and it are known to the acerulae are produced in and this pathogen is also known to produce ascostroma, which in turn contains the sexual ascocarp, and the asci, which are ovoid in shape, while the ascospores, which are 1 to 3 septate, elliptical, hyaline, and yellowish in nature. And these ascospores responsible for the spread of disease during the off season. Windborne conidia are responsible for the secondary spread. Sour orange, rough lemon, as well as the pomelo are highly susceptible to this disease. The young leaves are affected up to 100%. When young leaves are severely affected when 100% relative humidity and the temperature of 16 to 23 degree are prevailing. And leaf wetness is also responsible for severe infections. And then to manage this disease, collect and completely destroy those infected leaves, twigs, as well as the fruits to reduce the inoculum density. And in the standing crop, you have to apply copper oxychloride at the rate of 0.3% or Bordeaux mixture at the rate of 0.1%, or difoluton or chlorothalonil, etc. can be used for managing this disease. And these chemicals may be applied for 2 times at a fortnightly interval so that the disease got reduced in the living crop. And then next one is the Ganoderma root rot, and this disease is very severe, especially in the sweet orange and acid lime, and it produces white thick mycelial strands or the fungal growth on the barks of the roots, and these white strands of the fungi that become very dark, and the affected wood becomes soft and discolored, and it ultimately results in drying of the above ground parts and branches. And during the rainy season, at the base of the infected plants during the rainy season, the fungal pathogen reduced to known to produce woody brackets or fructifications. This is the typical fructifications produced by the pathogen Ganoderma lucidum on the infected plants. And how the disease spreads? The disease mainly spreads through the rhizomorph of the rhizomorph. Rhizomorph is the congregation of mycelia which form a scissor-shaped structure which pierces into the plant and causes the infection. The wind and waterborne basidiospores are also responsible for further spread. Severely infected plants or affected portions should be removed, and the soil should be mixed with the sulfur at the rate of 0.1 or 1 kg per tree up, and enriching the soil base with the green manures that will be helpful in reducing the inoculum density. Around the affected plants, you should form the isolation trench of 30 centimeter wide and 60 to 90 centimeter depth at a distance of 2.5 to 3 meter from the stem base. That would be helpful in isolating the infected plants from that of other healthy plants, so that spread could be avoided. And then drench at the base of affected plants with the Aureofungin at the rate of 0.3% or Bodo mixture 1%. That is advisable to reduce the disease incidence. And the next important disease is anthracnose, or withering of tip or dieback. In most of the cases, the anthracnose pathogen is known to produce some deep-seated lesions at the base of petiole or on the stem, and when the infection become very severe, it causes girdling symptom, and that may be responsible for formation of dieback. So the dieback symptoms with acerulae on the is found on the infected plant's stem region, yellowing, complete drooping and dropping of leaves from the affected plants are next indications. And then formation of reddish brown sunken lesions on the fruits are also characteristics of this pathogen, and at the center of these lesions there would be the formation of acerulae. The dark brown spot with grayish white center along with the numerous acerulae are noticed on the leaves. So, as we discussed, similar type of deep-seated lesions formed on the petiole as well as on the stem and as well as on the fruit. So, this is the typical symptoms. That is circular in nature. This is dark black sunken spots. A later extent become gray center and a dark brown margin. Similar type of deep-seated lesions that are formed on the fruits also. And this is the conidia. So the acerulae, which in turn contains conidiophore, conidia and setae. The conidia here, which are single-celled, oblong in nature with oil globules. And the primary spread is mainly through the dormant mycelium in the dead twigs or in the branches, and the secondary spread is through windborne conidia. Acid lime in the northern India is highly susceptible to this disease. Melta orange and grapefruits are also susceptible to this disease. Besides, the nitrogen deficiency and is also responsible for initiation or spread of this disease, are responsible for the infection, and the infection will be much high during the months of July to January. So in order to manage the disease, prune out the affected twigs, and then those cut ends should be completely protected with the Bodo paste, or foliar spraying of carbendazim at the rate of 0.1% or captopril at the rate of 0.2% are also advised. And then pink disease, which is caused by Pellicularia salmonicolor or Cortysium salmonicolor, which affects during the monsoon period. And the infection that is responsible for formation of whitish or pinkish fungal growth on the affected portions. Besides, it also produces slight gumming at the early stage of infection. In the infected plants, the leaves in the above point of infection, the leaves present at the on the infected plants just above the point of infection turns yellow and completely dry and fall off. Upon the affected portions, you can see the white silvery thin film with pinkish pustules. The bark of the affected portions or affected plants may become dry and develop some longitudinal cracks, and the internal wood turn into dark brown in color due to the infection. This is the white or mycelial growth with turn into pinkish at the time of sporulation. This is the longitudinal cracks developed on the trees, and the fungus is known to produce mycelium which are hyaline in nature, thin-walled and sparsely septated. The pustules are white or pink or orange red in color, and the conidia, which are thin, angular and single-celled, and it also known to produce the sexual spores, basidiospores. Some of the crops like cashew, cocoa, coffee, jack, mango, etc. may also act as a alternate host for this particular pathogen. And the windborne conidia and rainwater is responsible for the secondary spread, while the dormant mycelium present in the infected or affected portions are responsible for the primary spread. The vigorous trees are more susceptible, and the trees with the diplodia infection is also highly susceptible for the further infection by this Pellicularia salmonicola. And then in the affected plant, prune out the affected portions and then apply Bordeaux paste to protect the cutians. Foliar application of Bordeaux mixture 0.1%, 1% or lime sulfur is also advisable to reduce the incidence. Sooty mold, we have studied in many crops about the sooty mold. These are all the blackish encrustation or the fungal growth on the superficial growth on the surface of the leaves, as well as on the tender portions or tender parts of the plants, which are all the fungus which grown by utilizing the salivary secretion of the insect pests, especially the sucking insects. And this incrustation, which covers on the leaf, which reduces the photosynthetic activity or photosynthetic ratio, and ultimately responsible for raw productivity. Similarly, the formation of black powdery coating on the fruits that reduces the market value. This is the typical encrustation of fungal encrustation over the surface of the leaf and surface of the fruits. And this is formed by the saprophytic fungi Capmodium citri, which produces different types of spores like Coniothecium, Brachysporium, Trichothecium, type of Conidia, as well as Ascospores. And to manage this disease, as we learnt in many other crops, here also application of 5% starch, which is advisable when the starch are solidified or dried, then that forms the flakes on the surface of the leaf, and these flakes may fall off along with the powdery coating formed on the leaf or fruit. Or application of carbendazim at 0.1% is also effective against these sort of fungi, which is forming a superficial encrustation. Besides, spraying of some insecticides are also recommended, because that the salivary secretions of the insects that serves as the feed material for the development of Capnodium, and hence you need to control the insect population also by applying the pesticides which are effective against the sucking pest, like methyl dimethoate or dimethoate, etc. Then citrus canker or bark eruption, which are all first reported in England during 1933, but in India, especially in the Punjab, in the state of Punjab, it was reported in 1940. Here, the acid lime is highly susceptible to this disease. In the case of leaf canker, it produces small, circular, watery, yellow translucent spots with raised convex surface, and this is caused by the bacteria, and this spot later turn into white or grayish with rupture the center and giving a rough, hard, corky growth, and top of the corky growth may show a crater like appearance. Crater means the mouth of volcano that is opening with a shallow depression. So such type of appearance will be there above at the top of corky growth formed on the leaf as well as on fruits. And this disease is caused by the pathogen Xanthomonas axinopodis pathovarcitri. So the pathogen is known to cause symptoms on all parts of the plant, including leaves, twigs, fruits, thorns, etc. And in cankers, when the affected leaves become completely distorted, and the lesions are typically raised, rough, brown pustules, and having the crater like top, especially in this particular corky growth you could see, surrounded by the yellow halo which appears on both the surface of the leaves.
And with the similar type of raised corky growth may also be seen on the outer rinds of fruits also. In the immature fruits, you can see the formation of yellow halo, while you cannot observe the yellow halo in the ripened fruit. And this is the typical raised bacterial cankers on the stem as well as on the thorns. And this is the typical symptom formed on the fruit. As we discussed, in the immature fruits you can see the yellow halo surrounding the point of infection, while you cannot see the yellow halo in the ripened fruit. And the causal organism, that is Xanthomonas axinopodis variety citri, which is a gram-negative rod-shaped bacteria with a single polar flagella. It is also known to produce some yellow color pigments. The closer planting and the injuries caused by the thorns and leaf miner infestation, the as shown in this picture, the leaf miner infestations all are responsible for, are all the favorable factors for the establishment of this disease. And the acid lime, lemon, as well as the grapefruit are highly susceptible to this disease, and this disease will be more severe when the temperature ranges from 20 to 25 35 degrees Celsius are prevailing in the presence of moisture. And to manage this disease better, grow the some of the resistant cultivars like rangpur lime or rough lemon, etc. And then collection, destruction of infected leaves, plant debris, which serves as the source of inoculum. And then collect the seedlings from disease free nurseries, and prune out all those infected plant pots, and protect the cut ends with Bordeaux mixture or Bordeaux paste before the and this has to be done before the onset of the monsoon. And similarly prevent or control the infestation by the leaf miner, so indirectly you could control the entry points for the bacteria, and hence you can avoid the spread of the disease. And in severely affected plants, you can go for spraying with the streptomycin sulfate at the rate of 100 ppm mixed with 0.25% of copper oxychloride solutions, and this has to be applied for 3 times at 15 days interval, effective against the bacterial infection. The next important disease is citrus tristissa or quick decline disease. It was first reported in Italy, while now, but it is distributed throughout the world. The disease had practically wiped out the citrus plantations in many of the countries like Argentina, Brazil, and Uruguay. The lime is highly susceptible to this viral disease. And sour orange, pomelo, or lemon are highly susceptible rootstock for the viral infection, while the trifoliate orange or mandarin, rough lemon, etc. are the resistant rootstocks against this viral infection. And similarly, the sweet orange, mandarin, tangelo, and grapefruit are susceptible scions. And this causal organism is Citrus virus. When the adult tree of such a combination is affected, the affected tree, the leaves of the affected tree turn into yellow, and the entire plants will wilt rapidly and dies within a few years of infection. And this is the typical quick wilt or quick decline affected mandarin orange. And the infection of virus disease on the plant that shows development of venal flecks on the surface of the leaf, stunted growth of affected plants, and when you remove the bark of affected plant, you could see numerous pitting on the stem of affected plants. Ultimately, their roots become decayed due to infection, and the twigs showing the dieback symptom. The affected trees may die within a short period, and the fruits, if it is produced, very small in size and unviable. And then next, it also produces seedling yellows disease, that is self-rooted trees of sour orange, lemon, etc. are affected by this disease after being infected with the citrus tristissa virus, severe strains. And these trees become affected trees become yellow and showing the dieback symptom. If the affected trees are grafted into resistant rootstock, they soon will recover. And this is the typical venal flex and yellowing of leaves due to infection by the Citrus virus. Grapefruit, buntan, here we have studied this earlier, that is grapefruit, buntan and its relatives like tangelo, tanger, sweet orange, etc. are susceptible, and the infected leaves, venal flex and yellowing on the leaves, and numerous stem pitting, that is due to the infection by this virus pathogen on the tree trunk or on the stems, and the affected trees become very dwarfed or stunted in growth with less vigor. And in the in the tree showing some stem pitting symptom, you could see the profused flowering, but the formed flowers will be very small, fruits will be very small in size and irregularly shaped. There may be a development of oily spots or brown spots with gumming on these affected on the fruits that formed in the severely affected trees. The causal organism is the severe strain of Citrus virus, stem pitting virus, and this is the oily spot that developed on the affected fruits. And this Citrus virus, which is a long, flexuous and rod-shaped virus, and which is transmitted by the citrus aphid Toxoptera citricidis. Besides, the Cuscuta, which is a phanerogamic parasite, which is a daughter, nicknamed as daughter, is also responsible for transmitting this virus from infected to some of the healthy trees. This is the typical shape of the virus, flexuous rod shaped, and this is the transmitting vector, that is Toxoptera citricidis aphid. And to manage this disease, avoid using the susceptible rootstock, that is rootstocks from the grapefruits or sour orange, etc. Use the resistant rootstocks like collected from the trifoliate orange or mandarin. The pre-immunized seedlings or the cross protection process are helpful in reducing the disease incidence. Such seedlings are called pials, 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 pre-immunized acid lime seedlings, that is immunization of the seedlings with the mild strain of citrus virus, which gives resistance against the infection by severe strains. Cyan routing technique is also be helpful, and avoid control the insect vectors so that the transmission or further spread of the disease are controlled. And then Huanglong Bing, or citrus greening, is another important disease, and it has become serious nowadays. And this disease affected plants, the leaves of this disease affected plants are become stunted with only sparse foliation, and dieback of the twigs are also common, and green fruits with some yellow patches are common in the affected trees. And chlorosis of leaves due to this disease establishment, which resembles the zinc deficiency in the normal crop. The twigs that become upright in position with the smaller leaves. The fruits are very small and lopsided with some curved columella, and the sun exposed side of the fruits turn into fully orange in color, while the unexposed area still that remain green in color. The fruit juice quality from the affected fruits are very low, with high soluble solids and acidity, and in the affected fruits the seeds are very poorly developed, and which become colored, dark colored or aborted. This is the stunted trees due to severe infection ultimately lead to heavy leaf drop. The off-season flushing and blooming are common in the affected trees. The few fibrous roots or new roots may be formed in the affected trees, since majority of the roots got decayed, and ultimately resulting in development of dieback symptom. This is the citrus greening symptom on the leaves, which produces blotchy mottle type of symptom in between the intervenal portions. Intervenal portions. These symptoms may not appear for the first 2 years or more, and later this symptom will be the common. This picture clearly shows the difference between citrus greening affected leaves and the healthy leaves. The secondary symptoms are reduced leaf size, and then erect or upright leaves and with chlorotic symptoms which are similar to that of the zinc as well as iron deficiencies. This is the fruits, green fruits affected by the Hunglongbing disease. The sun exposed side turn into orange in color while the unexposed area remains still green, and this is the lopsided fruit with the thickened curved columella which reduces the quality of the fruits. The causal agent, which is a phytoplasma, that is Candidatus libribacter asiaticus, which is a phloem limited fastidious bacteria and got transmitted by the Asian citrus psyllids, that is Diaphorina citri. This is the psyllid Diaphorina citri, which transmits the phytoplasma and causing the Honglongbing symptoms. And the infection that spreads from the infected budwood by when the budwoods are collected from infected plants, and it got transmitted by the insect vector, that is Citrusilla diaphorina citri. The infected plants act as the primary source of inoculum, and the secondary spread is mainly through the insect vector. So remove the affected portions, use disease-free buds collected from the disease-free area, or the disease-free resistant rootstocks can be used for developing the seedlings. In case of severe infection, you can apply monocrotophos at the rate of 0.05% to control the insect vector. Application of tetracycline at the rate of 500 ppm at 15 days interval will be helpful in remissioning of the symptom, but the symptom will occur at the later stage. This is not the permanent control measure. And in citrus sorosis or California scaly bark. This is another important disease which produces small, elongated, light areas or the regions in between the venal portions, and besides it also produces vein flucking on the surface of the leaves, and upon the bars the superficial area of the bars may peel off, and then profuse exudation may occur, and sparse foliage and dieback are the associated symptom due to infection by the citrus sorosis virus. There are different strains, that is A and B, and this strain A is a severe strain, and it is a RNA virus and got transmitted by grafting or natural spread, possibly by the Alpidium brassicae, which transmits. The other ophioviruses are also transmitting this citrus sorosis virus. And budwood certification that helps in reducing the disease incidence. Prune out the affected area, which will helpful in reducing the further spread by scraping out of affected barks and treat it with the potassium permanganate at the rate of 1%, which will helpful in reducing the incidence and as well as reducing the further spread by reducing the inoculum potential. And the next one is citrus exocortis, otherwise it is called as a scaly butt. And this exo means outside, cortis means the protecting bark. Outside the bark there is an infection, that is mentioned as citrus exocortis, and this trifoliate lemon, Rangpur lime and then citrons are highly susceptible to this disease, and the infection that leads to development of cracking and scaling of bark, and besides it develops narrow vertical stripes from the soil line up to the bud union region, and it also spreads downwards to the root region also. The bark of the affected plants become completely dead and dried off, and the affected plants are stunted in growth, and it reduces the growth of rootstocks, especially in the trifoliate orange. This is the scaling of bark, especially on Poncirus. The causal agent is the viroid, that is Exocortis viroid, which is a RNA without protein coat with the 371 nucleic acids with the molecular weight of 1,11,000. Rangpur lime, citrus oranges, citron, trifoliate orange are highly susceptible, and it spreads through the infected budwood as well as contaminated tools. But there is no vector, there is no seed transmission is reported for this Exocortis viroid. And for managing this disease, remove the affected parts and use the tolerant root stocks like rough lemon, Cleopatra mandarin, sweet orange, and sour orange, and sterilization of the tools used for budding or some intercultural operations with trisodium phosphate or soap solution. Completely disinfect those tools and then use it in the field condition to avoid spread from the infected to that of the healthy plants. And the next one is the menace caused by the giant mistletoe, that is the growth of loranthus, which establishes a parasitic relationship on the trees, and it develops completely well emerged leaves and colorful fruits, and these colorful fruits are get attracted by the birds which eaten away these fruits, and then through the fecal matter the seeds are get disseminated from the parasitized tree to the healthy trees. This is the colorful berries developed by the loranthus, and the birds which eats the fruits, colorful fruits, and through the fecal matter the seeds are get disseminated from the parasitized plant to others. And the removal of the parasitized branches are highly advisable. So slay off or cut off the affected branches, protect the affected portion, cut portions with the bodo mixture or any other protective fungicides is highly recommended.